Hello, biologists. Welcome to lecture number 2, which is all about protists. Protists are incredible. They're grouped into 6 super kingdoms. Inside of each of those 6 super kingdoms are multiple kingdoms, and then each is believed to be a monophyletic group, but how they relate to each other, we don't really know, and we can see that on this phylogenetic tree. We know they all have the same common ancestor that leads to these eukaryotic supergroups, but we don't know where they branch off from each other. So that's where these dotted lines exist. We're pretty firmly believing in these lines. We feel like these associations are good, but these dotted lines are kind of a question mark. So we don't know who arose first, but we do know they all arose and they're all related to each other and they all have really interesting characteristics. So let's get into each group. In general, there's about 100,000 protists. They are all living eukaryotes, so they all have membrane-bound organelles. They're at least a cell, some are huge and multicellular. And what unites them is that they're not animals, they're not plants, and they're not fungi. In general, we use a term when we're talking about them. If they seem like plants, like this kelp here, and they do photosynthesis, then we call them algae. But if they don't do photosynthesis, we don't use the term algae. So the word algae will be used. It just means that they have chloroplasts and they are getting sugars from sunlight. So we also have some really interesting diseases that are caused by protists. I put these first because we want to think about humans and we want to think about public health. But I wanted to make sure that we discussed the grossness first, and then I'm not going to focus on their disease attributes later. So 3 of the big ones that protists cause are malaria, which is a huge impact on humanity. There's about 263 million new cases per year. That was from 2023, which is the most recent data. And then in 2022, there were 608,000 deaths. So pretty incredible. Here is your plasmodium. You can see it inside of human blood. These are red blood cells. So this is the protist inside of someone's blood. We'll talk about it a little bit more, but we're not going to do a major deep dive into disease. The next one is Giardia. There's only one species, so Giardia lamblia. The trick to pronouncing Latin names is just say every vowel and say it with confidence. So I sometimes stumble, forgive me, but just go Giardia. So every vowel. Lambelia. So Giardia lambelia. There's about 1 million cases in the US per year. It's rarely fatal, but what it does do is it causes really horrible diarrhea. And the dehydration can kill you, but if you get on fluids and go to the hospital, it's not usually fatal. Here's Giardia. It's got these beautiful little flagella. We also have sleeping sickness and Chagas disease. These are caused by trypanosomas. There's about 6 to 7 million cases yearly, and forgive the lack of the L there, about 12,000 deaths. Moving on from the disease-causing groups, we're going to go through each of the supergroups individually. So our first supergroup is Archaeplastida, and this is going to have the red algae, the Rhodophyta. Rhodophyta is the red algae. There's about 5,000 species of red algae. They're mostly marine. They're called red algae because they look red. They look red because they contain these different pigments that they use for photosynthesis. And those pigments reflect red light. There's 2 cell walls, and the components of those cell walls are economically important for us. We use the components of those cell walls to make agar, to make carrageenan. This is also the family that nori seaweed is in, so we eat these items often. Carrageenan is a stabilizer. It's in milk, it's in yogurt, it's in non-dairy creamers like oat milk, soy milk, almond milk. It's also added to things that have been mashed up and put back together. So deli meats like sliced turkey, that often has carrageenan in it. I went downstairs and I looked at my cheese and my tortillas, and both of those that I eat pretty much every day they contain carrageenan. So carrageenan, it is a byproduct of the cell walls of a red algae. So really common. We also have the chlorophyta. So rhodophyta, chlorophyta. Chlorophyta are the green algae. They're subdivided into two groups. The chlorophyte group, which is going to be the true green algae, and the charophytes, those are going to be the more ancestral. These are charophytes right here. This is the genus Chara. And we think that this is going to be similar to the most common recent ancestor of land plants. So we think that it is more ancestral. It's the more historic type of plant that existed and was more like the ancestor that gave rise to what we think of as land plants, which we'll go into in detail in 2 weeks or so. In the charophytes, we have the genus Chara, which I showed you on the last slide. Then we have Spirogyra. Spirogyra is this. This makes like a pond scum, but if you look at it under the microscope, its chloroplasts are long and skinny, and they swirl around the inside of the cell. So it's spirally, that's Spirogyra. This is often included in greens powder. So if you drink a green drink or you make smoothies with a green powder, it often has Spirogyra in it. Then we have Clostridium. Clostridium is this beautiful boomerang-shaped algae. So it's green, it's photosynthetic, really, really gorgeous in the scope. And then we have Desmodales. Here's an example. Looks like a beautiful little starflake of a green algae. So these are all green algae in the charophyte group. Then we have the true green algae, the chlorophytes. We think that land plants were derived from this group. They're interesting because they have a cell wall that contains cellulose, which is what we see in our land plants. They mostly use chlorophyll A and B, which is true about land plants, and they store their excess food as starch, which is true about land plants. They're really common in plankton, so they're really important to the food web. Everybody in the ocean eats plankton or eats somebody that eats plankton, so they're really important as a base food source. This is the theoretical chlorophyte, so it's going to be a single cell with flagella, and it has modified chloroplast, it has nucleus, it has Golgi bodies, it has mitochondria, and we have a chlorophyte that looks just like this. So this is Chlamydomonas. Chlamydomonas has the same shape. It has those two flagella on top. And inside of Chlamydomonas, we have the nucleus and the big chloroplast used for photosynthesis, little mitochondria, Golgi body. Chlamydomonas are tiny, and they're found in freshwater ponds, and they swim around, and we have found fossil evidence for them dating back 1 billion years. So it's pretty cool. They haven't changed much since then. Really beautiful green algae that you find in water. Here's another chlorophyte. This is Volvox. Volvox is cool because if you were to look at each of these little green dots, each one of the dots looks like a Chlamydomonas, but they organize into a colony. And the colony makes a hollow sphere. And when one of these cells starts to reproduce, it creates its own colony inside of the sphere. So these cells, when they eventually die, will release these new colonies. And so this is our first colonial algae. It's pretty cool. And the whole sphere itself is wiggling its flagella, and it twirls through its environment. Really neat. And it has some interesting specialized cells for reproduction that you can read about online if you want to hear about cool algae sex. We also have some edible green algae. This is the Caloptera. It is one cell. This entire thing, like this string of grapes, is one cell with many nuclei. There are not cell walls dividing the nuclei, so we call it cenocytic. And this is delicious, edible. It's some of the biggest cells in the world. So this is all one cell here. This strip is one cell. Here you can see it at a fish market in the Philippines. These are also Calyptera, and they're also one cell, so those are pretty cool. It's also an invasive species. So any Mediterranean climates like the coast of California, this is actually a problem. So we should be harvesting it and eating it instead of letting it get a foothold and growing. Now we're into our second supergroup. This is the amoebozoa. This is a huge group, and what they do is they used lobed pseudopodia. We're going to have another group that uses pseudopodia, but they aren't round like this. Here's a lobed pseudopodia. So this is a large projection, and what they do is they push their cytoplasm into it so it fills kind of like a water balloon, and then they contract the rest of the body into the part that is swollen, and they crawl and creep around that way using their pseudopodia. They also use their pseudopodia to eat. So they do phagocytosis. They engulf organisms in those pseudopodia and break them down. There's a type of entoamoeba that affects roughly 35 to 50 million people worldwide. It's called amoebic dysentery, and it kills about 55 people per year. And as you would think with dysentery, it mostly is uncontrollable diarrhea and water loss and vomiting. And so these little amoeba can be problematic, but that's one species in this larger group, of which we have two types of amoeba we're going to talk about, and then slime molds. So here's our beautiful slime mold. This is a Physarum, and it's showing you that it solved a maze. What they do is they put a food source on each side, and then they put this giant amoeba-like slime mold in it, and it projects its pseudopodia through, and it finds the fastest route to the food source, and it is smart, which is fun. What's really neat is these little slime molds, it's basically a cell using pseudopodia to move around, so that's why it's in this supergroup of amoebozoa. It doesn't have neurons, it doesn't have a brain, but it's still smart enough to determine the quickest path between two routes. So slime molds are really incredible. This is the slime mold life cycle. We're going to start with little spores. Those spores are haploid. Haploid spores will fuse together. As they fuse together, we call it fertilization. When they fuse together, they create a diploid zygote. This is really similar to us. And that diploid zygote will then do mitosis and grow. But what's different is this mitosis does not do cytokinesis. So this is a single cell with tons of nuclei, hundreds, maybe even thousands of nuclei. And so now I have this massive big cell, and it goes roaming around in its feeding stage. So this is what it looks like in real life. These live in our area. I see them on campus every fall. And this will crawl around ingesting tiny little invertebrates because it's a hunter, and it ingests them with its pseudopodia. And when it's ready, it will find a place to sit. It will produce these little egg structures. It will elevate the egg structure. And inside that egg structure, it will do meiosis to produce spores, and the whole process will start again. These have an annual life cycle, so they're in their eating phase in the fall, and before winter, they release their spores. Here's our other supergroup, our third. It's Opisthoconta. So Opisthoconta. This is the group that we're in. So this is our favorite group because animals exist in this group. And it's named for having certain cells with a single posterior flagellum. Think about human sperm. That's why we're in this group. There are a couple different types. China flagellates are unicellular or colonial. Here I've shown you one that's in a colony. So it's got these projections, and then you can see the long single posterior flagellum coming out past those projections. We think this is what the ancestors of animals look like. In the second group in this family, there are parasites, but they're mostly parasites on fish. And so we're not going to go deep dive into that. But if you love fish, you should look up this type of family. It's really interesting. Our next supergroup is Rhizara. Rhizaria, pronounce every vowel, Rhizaria. This one is also using pseudopodia, but instead of big lobed pseudopodia, it uses thin thread-like or hair-like pseudopodia. We can see it in this image here. Each one of these little hairs coming out is a pseudopodia. And so those pseudopodia will grab food and pull them back and ingest them with phagocytosis. This family has really cool organisms in it. They create an armor-like structure, and it's called a test. Now, the test can be calcium carbonate, or it can be silica, silicon-based, or it can be strontium salts. I'm going to show you images of them coming up. They're really beautiful. All of these family in the Rhizaria are really important to our nutrient cycling systems, specifically carbon and nitrogen. They're actually really important to climate change because as they absorb carbon in the foods that they're eating, they incorporate it into their body. And when they die, their little bodies fall to the bottom of the ocean and no one eats them. And so they sequester carbon out of our atmosphere. So they're decreasing our carbon load, which is important because we have too much carbon dioxide in our atmosphere. In this group, we have two families. We have foramids. I'm not going to say the full name, but we call them forams. Here are our forams. These forams have tests that are made of calcium carbonate. So really beautiful, spirally, almost look like a snail shell or a conch shell. Absolutely gorgeous. And then we have radiolarians. So here are your radiolarians on the right. They also create tests, but their tests are made of silica, which is glass. So they are made of little beautiful glass shells, really intricate. Look at this one here that's kind of X-shaped. Absolutely gorgeous. Our, I think, fourth supergroup is Chromalveolata. So just say it with confidence. These are the alveololates, and they're called this, think alveolus in like your lungs, because there's these little air sacs right inside of their membranes being shown with the red circles. So those little alveolates are important for something, but we don't know what. Their function is unknown, the alveolus. And so the Chromalveolata are all of the protists that have these little air spaces. In them, we have dinoflagellates, apicomplexan, and ciliates. So really neat. Dinoflagellates are a type of alveolate, and they are really morphologically diverse, but they are all photosynthetic or heterotrophic, and then a fun word for us, mixotrophic. So there's going to be dinoflagellates that sometimes do photosynthesis, and they sometimes eat other organisms. So they do both. They are mixotrophic. They're mixing it up. Each of them is encased in an interlocking plate of cellulose. We find cellulose in plants, and they are common in plankton, so they're really important for our food webs in the marine environments. Some are bioluminescent. I have a video about bioluminescence that I'm going to link to on the Blackboard site. It's really cool, so please check it out. It's going to explain what's happening here, and you're going to see a local example from the Pacific Northwest. Some of these also cause toxic red tides, which will create fish kills. And these toxic red tides can be poisonous to other organisms if they get in the water. In the alveolates, we also have apicomplexans. Apicomplexans are named that because they have an apical complex. We think it's a modified chloroplast. We're not totally sure. All of them are parasitic, so they're not doing photosynthesis with their apical complex. And the one that we care about most as a human is Plasmodium that causes malaria, which affects millions and millions of people. Here is a Plasmodium life cycle. What's neat about the Plasmodium life cycle is there's two hosts. There's a mosquito host and there's a human host. And inside of the human host, there's two stages. So here's a mosquito. It bit this human. And as it bit this human, it injected the parasite, the protist, into this human's bloodstream. Fun fact, only female mosquitoes bite people. They need the blood to produce eggs. The males don't bite people because they don't make eggs. So this female mosquito bit this human and injected a little bit of the protist into this human. That protist will go to the liver. It will infect the liver, and it will increase its numbers until it finally moves toward the blood. The protist will then stay in the human's blood all of their life. And as it lives in the blood, it will periodically produce gametes. And so here you have your gametocytes. Those gametes are what infect mosquitoes. So this mosquito, if it was unaffected, bit this human, and now it's infected. So in the mosquito, the life cycle of the plasmodium then goes into the zygotic form. So those little gametes will fuse, they'll become a zygote, the zygote will do mitosis, and it will produce these little tiny bits of itself that can then infect another human. So we have two different hosts, two different stages, mitosis and meiosis occurring, a really complex and unfortunate for us because malaria kills hundreds of thousands of people, and majority of them that die are under the age 5. And so it's a really awful disease. In this group, we also have ciliates. Ciliates have cilia. So instead of a long tail coming out the back or the sides, it's little tiny tails all over the body. So here you can see the cilia really well. In the paramecium, the cilia is all over the side. I don't know if you can see it on your computer, but it's covered in cilia. You can see it on the edges there. And they beat the cilia in unison, and they swirl through the water that they live in. They're really cool. Both paramecia and tetrahymena are important model organisms. Model organisms are things that we understand really well and that we can grow pretty easily. And so we use them in lab. So we use paramecium and tetrahymena in lab often. Tetrahymena you might have heard before because that was the model organism we used to discover telomeres and telomerase. So remember, telomeres are the little caps on the ends of your chromosomes, and telomerase is the enzyme that extends the telomeres. Telomeres tend to be related to aging. All right, here's a paramecia life cycle. This is kind of wild. They have two different types of nuclei. They have micronuclei that are used for sexual reproduction, and they have macronuclei that are used for asexual reproduction. The micronuclei are not transcribed, and the macronuclei are transcribed. The macronuclei are created from the micronuclei. So here we are. We have a paramecium. It's going to have sex with this other paramecium. To do that, they join their bodies together, so they make a little bridge of cytoplasm. Each of them have their two different types of nuclei. They're going to take their micronucleus and they're going to do meiosis with it, producing 4 of them, but only one survives, and we're going to see a trend with this throughout the animal kingdom. So of the micronucleus that survives, it's going to divide again by mitosis. They're going to swap micronuclei. Those micronuclei are then going to fuse, creating a diploid micronucleus. That diploid micronucleus is going to do mitosis. It's dividing. And as it divides, the original macronucleus disappears, and the micronucleus will become the new macronucleus. And then each paramecium that now has 4 micronuclei will divide into 4 whole new paramecia. Each of those micronuclei is different than the parental micronuclei because it did fertilization with the micronucleus of the partner that it paired to. So kind of a wild life cycle. Your book does a good job explaining this as well. Here we have our straminopiles. Our straminopiles are diatoms, brown algae, golden algae, and oomycetes. You'd say both O's. You could say oomycetes, but remember we want to pronounce every vowel. So oomycetes. They all have a feathery flagella. Sometimes it's described as hairy or super divided, but one of them will be hairy like this. And this is a weird group. There's all sorts of weird things. Let's look at the diatoms first. Here are your diatoms. They're unicellular, they're photosynthetic, they have cell walls of silica, so this is glass. They are big in plankton, so they're important for our ecosystems. They're also used in soundproofing and polishing and filters. So important diatomaceous earth. Diatomaceous because it contains diatoms. Golden algae is cool. It's golden because it has carotenoids in it. Here's your little golden carotenoids. It's mostly unicellular, but sometimes it's colonial. This is a colonial one right here. I thought it was really beautiful. Mostly freshwater, not well studied or understood. And then our brown algae, which is maybe my favorite because it's so weird. You've probably seen kelp before. Kelp is huge. It looks like a plant. It can grow 200 feet tall. It makes forests. If you look at the individual leaves, they look like a leaf and they have a stem, and there's like a root structure at the base that holds it in place, but it's not a plant. It's a protist. It's a colonial protist. And that kind of blows my mind that a protist can be so big and so plant-like. And so that's why I love it so much. Majority of the seaweed that we eat, and seaweed is delicious and so good for you, are brown algae, except for that nori. The nori was a red algae. Here's your kelp life cycle. The big body of the kelp is called a sporophyte. Sporophytes make spores. It's diploid. It makes its spores in a structure called the sporangia. It will do meiosis. It'll create those spores. It releases them into the water. Those spores will germinate and they'll grow into what we call gametophytes. Gametophytes create gametes. There's a male and a female gametophyte. Each of those will produce a sperm or an egg, the gametes. The sperm and the egg will fertilize each other, creating a diploid zygote, and that diploid zygote will grow into the sporophyte again. So, beautiful process. Then we have our chrome aviolata. In this group is straminopiles, and in the straminopiles is the oomycetes that we mentioned earlier. Oomycetes are water molds. We used to think they were fungi because they have a filament, and the filament is used to uptake nutrients, but they're not. They're not photosynthetic, so they're not in the plant group, so we don't call them an algae. Instead, they are saprotrophs, which means that they digest dead material, so they're decomposers, and sometimes they're parasites. But they're not fungi, and one of the reasons they're not fungi is because their cell walls are cellulose, and fungi use a compound called chitin. So they have a different component in their cell walls. Here is a mayfly larva. This is its head. These are where its wings would be. This is its tail. And then all over it, these hair structures, that's the water mold decomposing it. So that's the saprotrophic life cycle, is decomposing dead things. Important for humanity, we have the potato blight in this group. So the potato blight, here you can say this to yourself. This is the cause of the Irish potato famine. It was in the 1800s. It was a mass starvation event. It killed millions. The population in Ireland went from 8.4 to 4.4 million. It wasn't that many deaths. About 2 million, 1 to 2, depending on your source, people left Ireland, and people that left Ireland because of the starvation event, most of them moved to North America. So if you have Irish lineage in your family, it very well may be because of this protist that your family moved. The population of Ireland has still not rebounded. It's currently only 7 million. This is what the potato blight looks like. This protist infects potatoes and creates all this necrotic material, and you can't eat this. It'll make you sick. It's decomposing. There's also many different types of blights that affect different types of plants. They're a huge cause of economic loss in agriculture. Then we have the supergroup excavata. They are asymmetrical. They're single-celled, and they have a groove in which they put parts of their body, which is pretty neat. It's called a feeding groove that's excavated, hence the name excavata. In this family, we have parasites such as Giardia that I'm showing you here. We used to think that Giardia didn't have mitochondria, but now we know that's not true. Instead, they have a mitosome, but it doesn't do cellular respiration. Instead, it does iron and sulfur metabolism. And so these organisms live in anaerobic environments. They're really interesting because they don't do full cellular respiration. Instead, they rely on glycolysis and alternative forms to create energy. So really bizarre little group. We also have euglenozoans in this. Some are parasites, some are heterotrophs, some are autotrophs, and some are mixotrophs. So really all variable in how they live their life. But what's cool about them is they have an eye spot. Here's this little red eye spot on Euglena, and Euglena can see, and it'll move toward or away from the light using its eye spot. Not a true eye, but a sensory structure, not an organ, because these are single-celled creatures, but a sensory organelle that can detect light. Euglena, they have these green chloroplasts all through them, so they can do photosynthesis, but they also eat, and so they're mixotropic. And then we have our trypanosoma, which is our sleeping sickness causing parasite that also causes Chagas disease. Here it is in human blood. Each one of these pink things is a human blood cell. So you can see that tiny little protists infecting human blood. And those are the protists. I hope you enjoyed that. Your textbook goes through each group in detail. Email me anytime and have a lovely day.